Hi, I'm Miss Bulla, and I am here today to teach you about eukaryotic cells, structures, and functions. Today you will learn the three main components of the eukaryotic cell: the secretory pathway of a protein, and the structures and functions of the organelles and key structures found in eukaryotic cells. There are three main components of the eukaryotic cell: the cell membrane, cytoplasm, and the nucleus. What are the internal structures of eukaryotic cells? Eukaryotic cells contain many organelles. And organelles are like little organs, and each one has a specific job to do. Our lungs help us breathe. Our heart pumps blood, which carries oxygen and glucose to each cell. Our pancreas releases insulin to control the amount of glucose in your blood. And all of the organelles have very specific functions, just like the organs in your body. First, let's take a look at animal cells. Eukaryotic cells include protista, fungi, plants, and animal cells. Let's begin our journey from the inside of an animal cell and look at the nucleus. The nucleus is made of 5 parts: the nuclear envelope, we also call that the nuclear membrane; the nucleolus, which is made up of DNA, RNA, and proteins; the genetic material, and sometimes we call it chromosomes, sometimes we call it chromatin or DNA; the nuclear pores, which are basically just little holes in the nuclear envelope; and the nucleoplasm, which is the cytoplasm of the nucleus. The nucleus. Can you identify the structures? The nuclear envelope, chromatin, nuclear pores. I have arrows pointing to 3 of them. There can be hundreds of those. The nucleolus, and the nucleoplasm. Remember, it's like the cytoplasm of a cell, but it's in the nucleus. Now, let's look at the functions of the nucleus and its parts. The main functions of the nucleus is to store and protect the genetic material or DNA of the organism. It is also able to direct the activities of the cell and tells the cell which type of cell it needs to be. For example, it could be a skin cell, or a bone cell, or even a neuron. The nuclear envelope, also called the nuclear membrane, is made of 2 phospholipid bilayers. You can see here how the outer layer and the inner layer are separated by space. The nuclear envelope is twice the layers of the cell membrane. The primary function of the nuclear envelope is to protect that genetic material inside. The function of the nucleolus is to make ribosome parts that will be assembled outside of the nucleus in the cytoplasm. It is not bound by a membrane and is basically a dense region of DNA and RNA and protein. Now this is a ribosome. It is made of 2 subunits. The nucleolus makes these subunits, but because they are so large, It ships them out individually through the nuclear pores. Ribosomes make proteins, and this is how they do it. The two ribosomal subunits, they come together in the cytoplasm of the cell. You can see how the messenger RNA strand slides through the subunits and meet up with the transcript RNA to make protein. Now, we haven't studied the cell cycle in our class yet, but you can see how there's chromosomes during the M phase, which is during mitosis. And then chromatin, if you look in the microscope, that's what you see, these loose strands of DNA. And then, of course, during the S phase, the DNA is going to be copied. Now, this illustration shows us the 3 stages or formats of genetic material. Nuclear pores are actually kind of complex, but for this class, you need to know that they allow for the passage of molecules through the nuclear envelope. We have already seen how ribosome subunits exit to make proteins in the cytoplasm, but other materials can pass through as well. Building blocks of DNA must enter as well as nutrients. It also allows waste to exit the nucleoplasm. Many higher eukaryotic cells have as many as 2,000 nuclear pore complexes in the nuclear membrane of each cell. Nucleoplasm is a highly gelatinous, sticky liquid that suspends the chromosomes and nucleolus and provides a medium for molecules to travel through. Now, let's move on to the next main part of the cell, cytoplasm. It provides a medium for the molecules to travel through and float in. And last but not least is the cell membrane, also known as the plasma membrane. This complex structure is a phospholipid bilayer embedded with proteins and bedazzled with carbohydrates and cholesterol. The main function, of course, is that it serves as a protective barrier from the external environment inside of the organism. But it also has the ability to choose what molecules can enter and exit. It is an amazing structure. Now, let's do a quick review. What are the 3 main components of the eukaryotic cell? I'll give you a moment to think. And if you said nucleus, cytoplasm, and cell membrane, ding, 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 you are correct. Great job. Give yourself a pat on the back. Now that we know the 3 main components of all eukaryotic cells, let's look at the organelles that make them so special and complex. The endoplasmic reticulum, also known as the ER. There are 2 types, the rough and the smooth ER. The rough is covered with ribosomes, so it's easy to identify. As we saw before, ribosomes are little protein factories. When the ribosomes are attached to the endoplasmic reticulum, the proteins travel through the ER and are modified or changed and then sent on their way to be packaged by another organelle. Functions of the smooth ER. By the way, this is Pebbles, my cat. The smooth ER makes and stores phospholipids for the cell membrane. It packages and connects molecules made in the rough ER to the cytoplasm and Golgi. It makes carbohydrates and steroid hormones, and it's responsible for the detoxification of medications and poisons. You'll find a lot of the smoothie R in organs such as the liver. The Golgi body, also called the Golgi apparatus. It can receive molecules from the ER to sort, modify, and package them into a vesicle for shipping. Now, we call the location of the journey of a protein the endomembrane system. And what we are going to look at is the pathway that a protein goes through as it's being made or synthesized. So the beginning of the journey starts with ribosomes that are made in the nucleolus. The ribosome exits through nuclear pores. They attach to the rough ER, and newly made protein is made on the ribosome and enters into the rough ER. Protein travels through rough ER and exits either through the smooth or the transitional ER and gets packaged into a little vesicle. It's like a little sack. Proteins exit the smooth ER and it travels through the cytosol and enters the Golgi. Golgi sorts, modifies, and packages proteins into the Golgi vesicle. And then the vesicle travels to the cell membrane and exits through the process of exocytosis. That is a lot of information. But you can look at the illustration and go through the steps. I've got them numbered. All right, here is a quick review. What is the pathway of a protein? All you have to do is just name the structures involved in the correct order. You will have to practice this several times to get this. And then briefly describe what happens along the way. So, it starts at the nucleolus. That's where the ribosome subunits are made. And then the ribosomes travel through the nuclear pore. Some of the ribosomes attach to the rough ER. Some of them are just freely floating around in the cytoplasm. The ones attached to the rough ER will make the proteins and either travel through the rough ER and then exit through the rough ER to go to the cytoplasm, or it can travel onto the smooth ER, which is connected to the rough ER, and they can be packaged by the membrane of the smooth ER, and then that will travel next to the Golgi. The Golgi sorts, modifies, and packages molecules to be shipped out of the cell, and then they will leave the Golgi in a little vesicle and go and attach to the cell membrane so that it can exit through exocytosis. So once again, nucleolus, nuclear pore, the rough ER, and then either to the cytoplasm or to the smooth ER. If it goes to the smooth ER, it will travel to the Golgi, and then it will go to the cell membrane to exit the cell. Now, let's look at organelles that store, clean up, and support and move the cell. The first one we will look at are called vacuoles. Cells can contain different types of vacuoles. Some of these sac-like membrane enclosed structures just hold water. Others may hold nutrients like protein, carbohydrates, and salts. Paramecium have vacuoles, and they are called contractile vacuoles that pump water for their movement. And here's a little video of how that works. Smaller vacuoles that are made from the membrane of the endoplasmic reticulum and the Golgi body are called vesicles. Vesicles store and carry materials from organelles and to the cell's surface. Vesicles store and carry materials between organelles and to the cell surface. Lysosomes. They contain enzymes that break down proteins, carbs, and lipids that can be recycled and reused later in the cell. Sometimes they are used to attack foreign invaders, like this white blood cell that contains many lysosomes. There are several diseases caused by lysosomes that fail to function properly. Usually in class, we like to call them the cleanup crew because they do contain those enzymes to break down waste in the cell. Organelles that offer support include two main types of cytoskeleton. They are called microtubules and microfilaments. Microfilaments are thread-like structures made up of proteins called actin. We see a lot of those in muscles. And then there's microtubules, which are hollow structures made up of proteins called tubulin. There is a third type of cytoskeleton called intermediate filament. It is a smaller fiber than microtubules, but larger than microfilaments. It provides structure in nerve and skin cells, especially the nails, claws, and hooves of animals. Not only does the cytoskeleton facilitate cellular movement and spans across the cytoplasm of a cell, acting as beams for support, but it also allows molecules to travel across from one part of the cell to another. So you could say that the cytoskeleton also acts as a roadway or bridge from one part of the cell to another. Quick review. Which organelles store, clean up, enable movement, and provide support for the cell? If you said vacuoles and vesicles store things, you are correct. Lysosomes clean up, and cytoskeleton is used for movement and support. Organelles that capture and release energy is our next category of organelles. Chloroplast and the mitochondrion are two organelles that capture and release energy. Ultimately, all energy comes from the sun. Without the sun, there would be no life on Earth. It is the driving force. Eukaryotes that contain chloroplast are plants, obviously, but also chloroplast is found in some protists, such as algae. And don't forget that Protista is one of the kingdoms of the eukaryotic domain. Photosynthesis converts solar energy to chemical energy stored in food, and this happens in the chloroplast. You may remember the photosynthesis equation from previous years in school. The organelle responsible for energy, the mighty mitochondrion. These fascinating and energy-packed organelles are found in almost every eukaryotic cell. They take basic ingredients in the cell and turn it into an energy molecule known as ATP, adenosine triphosphate, the molecule that eukaryotes use to do work. This work includes all of the chemical reactions that take place in an organism to keep it alive. And last but not least, cellular boundaries. While the cell's boundaries are not considered organelles, They are extremely important. There are two key structures, the cell membrane and the cell wall. Both the cell membrane and the cell wall are protective barriers, but cell walls also provide strength, support, and shape to the cell. Now here is a comparison of eukaryotes and prokaryotes. Eukaryotes all have a cell membrane. Some of them have cell walls. Two examples are plants and fungi. Prokaryotes, all of the bacterium, all have a cell membrane, but most prokaryotes have cell walls, almost all of them. Now this looks at the difference between the parts of the cell wall and the cell membrane. Sit and study it just for a moment. So the cell membrane obviously is made from a lipid bilayer, and you can see that same layer right here underneath. For the cell wall, you can see those microtubules and the microfilaments. There is cellulose and proteins embedded in here. And over here on the cell membrane, you can also see proteins embedded. So that is all of the structures and functions of eukaryotes. The three main components of the eukaryotic cell was the first thing that we learned. What were they? The cell membrane, the cytoplasm, and the nucleus. Good. We also learned the secretory pathway of a protein, and we learned the structures and functions of the organelles and the key structures found in eukaryotic cells. Thank you so much for joining me. I'm Deborah Bulla, and this has been Biology Made Easy.